So the basic definition, the common definition for cells is that a cell is the basic structure and functional unit of life. And in the organization of life, a group of cells that are similar in function form tissues, while a group of tissues similar form organs, and a group of organs that perform similar function will form systems. So the hierarchy is from cells forming tissues, tissues forming organs, organs forming systems. So we now have the various systems: the digestive system, the cardiovascular system, the skeletal system, and all. The human body generally originates from a single cell, and this single cell is called the zygote. When the sperm and the ovum come together, they fuse, and they fuse to form a unicellular organism known as the zygote. So we all are formed from this single unicellular organism called the zygote. And the zygote is the fusion of the sperm and the ovum, and this fusion gives us the zygote. You know, the sperm has 23 chromosomes, the ovum has 23 chromosomes. So when they fuse together, it gives us a diploid cell called the zygote. So as the fetus grows, the zygote undergoes cell division, leading to the development of specialized cells that share the same genetic makeup as the zygote. Now, so let's talk about the cell structure and the function. Each cell has a plasma membrane. This is the plasma membrane enclosing organelles in a watery fluid called cytosol. I told you under fluid that the fluid inside the cell, all these blue compartments represent the fluid. I told you the fluid inside the cell is called the cytosol. So everywhere you see blue, 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 these are fluid. Then the organelles rather, are swimming in this cytosol. So the cytosol and the organelles, excluding the nucleus, are called the cytoplasm. Now this is the nucleus. This purple is the nucleus. So if you exclude this nucleus, this fluid and all these other organelles make up the cytoplasm of the cell. So it's correct to say that the cell is made up of the plasma membrane, the cytoplasm, and the nucleus. So let's take the structure one after the other. So we are going to be starting with the plasma membrane. The plasma membrane is a phospholipid bilayer. This is a layer, this is another layer. All these round figures are the heads of the phospholipid bilayer, and the head is hydrophilic, meaning that it loves water. This head now is hydrophilic, while the tail is hydrophobic, meaning the tail hates water. So the plasma membrane, the phospholipid bilayer, are arranged in such a way that the tail are locked into each other, while the heads are on the opposite ends. So please remember, the plasma membrane is a phospholipid bilayer, and it has hydrophilic heads and hydrophobic tails. Then in this phospholipid bilayer, we have proteins and sugars embedded in the phospholipid bilayers, and these proteins and sugar function as channels, receptors, and enzymes. Then we also have cholesterol, and this cholesterol adds rigidity to the plasma membrane. So we have said that the plasma membrane is made up of the phospholipid bilayer, and proteins and sugars are embedded in them. Worthy of note is also that the cholesterol makes the plasma membrane rigid. So let's talk about the functions of the proteins that are found embedded in the plasma membrane. There are some proteins that extend through the membrane, meaning that this protein starts from the outer part of the membrane to the inner part of the membrane. Take a look at this protein I'm pointing the cursor on. This is a transmembrane protein. Transmembrane means it starts from the outside to the inside of the plasma membrane. This protein I'm pointing at, we can say this one is not a transmembrane, but if you see this one, this is a transmembrane. It starts from the outside of the plasma membrane to the inside of the plasma membrane. So what are the functions of the membrane proteins? The proteins that are transmembrane, that extend through the membrane, they act as channels, and these channels allows the passage of substances like electrolytes and non-lipid soluble molecules. Then some membrane proteins have carbohydrates on them, and these ones are for immunological identity of the cell. Some proteins also act as receptors for hormones and drugs. You know, your drugs act on most of the drugs act on receptors, and these receptors are just proteins on the plasma membrane. Then some of the proteins also act as enzymes. Now, so the function, what is the function of our plasma membrane? The plasma membrane is selectively permeable, meaning that it helps regulate the entry of substances into the cell and the exit of substances out and the exit of substances. This is the major function of the plasma membrane. It is selectively permeable. It controls or regulates how substances move in and out of the cell. So because of this function, we can say the cell has some certain transport mechanisms. We have the passive transport and we have the active transport. We have the active transport and the passive transport. The passive transport does not require energy, while the active transport requires energy. The passive transport, we have diffusion, osmosis, and facilitated diffusion. We have diffusion, osmosis, and facilitated diffusion. These are the three mechanisms under passive transport: diffusion, osmosis, and facilitated diffusion. You see, one of the things that can help you when you are reading, you can form what we call mnemonics. Mnemonics are just the things that you use to aid your memory. Mnemonics, we call them mnemonics. Take for instance, if I want to master the passive transport, I can say DOF, and the DOF means diffusion, osmosis, facilitated diffusion. So these are ways you can memorize things easily. Then for the active transport, it requires energy. The energy the cell uses is ATP, adenosine triphosphate. That's the energy that can be utilized by the cell. Adenosine triphosphate is the energy that can be utilized by the cell. So under active transport, we also have bulk transport like endocytosis, which can be divided into two: pinocytosis and phagocytosis. Pinocytosis means cell drinking, and phagocytosis means cell eating. Then we also have exocytosis. So let's take the diffusion. Okay, so diffusion is the movement of substances from a region of higher concentration to a region of lower concentration. So it means they are moving from high to low concentration, and energy is not involved. Let me give you a typical example of diffusion. If you stay in a corner of your room and spray a perfume, let's say another person is at the other end of your room, but you are in a corner and you sprayed perfume. in no time, maybe under 1 minute, that perfume will diffuse from your own hand to the other hand where the other person is saying, and the person will be able to perceive that. Okay, then the same thing, if you are on one side of your room and another person is on another side of your room and you fart, the person on the other side, if the fart is foul smelling, the person on the other side in no time will be able to sense that did you fart? So that's diffusion, because your gas will move from your hand, which is the region of higher concentration, to the hand of your roommate, which is the region of a lower concentration, and the person will be able to perceive it. So examples of substances that move in and out of the cell using diffusion, we have oxygen, carbon dioxide, fatty acids, steroids. Then I told you that there are some channels on the plasma membrane. So some substances also cross by diffusion through water-filled channels, some electrolytes. So what is facilitated diffusion? In this facilitated diffusion, we have substances moving through the plasma membrane by binding to carrier protein. So for these substances to move, they must bind to proteins on the plasma membrane. So this protein now turns. Let me explain what facilitated diffusion is. Let me use this diagram. Okay, consider this protein now. This protein, this is the upper part and this is the lower part. So if a substance binds here and this protein now turn upside down, you know that the substance that bind here will go inside the cell. Okay, so that's what happens. There is a substance binding here, then the protein now turns upside down to deliver the substance inside the cell or outside the cell. So that's what facilitated diffusion involves. It involves a carrier protein, and the substance binds to the carrier protein, then the carrier protein changes shape to deliver the substance on the other side. Osmosis is the last thing under the passive transport, and osmosis is just the movement of water. So what happens is that water moves down its concentration gradient. So if you have, but osmosis must involve a semi-permeable membrane. If you do not put semi-permeable membrane in osmosis, then you have not defined it. These are the two things: semi-permeable membrane, then it is the movement of water, the passive movement of water. These are the two things that make other things don't move in osmosis. It's just the water, and it requires a semi-permeable membrane. If you fart in one corner of your room, it does not require a semi-permeable membrane. It will just diffuse and the other person will be able to perceive it. The same thing when you spray your perfume, it does not require a semi-permeable membrane. The other person at the other end will be able to perceive it. But for water to move in osmosis, it needs a semi-permeable membrane. So water moves from a region of lower solute concentration to a region of higher solute concentration. So water moves from a region of dilute or diluted solution. You know, when we say something is diluted, it means that it has a higher percentage of water. So water moves from a diluted solution to a concentrated solution. That's why you see that. Okay, let me not go into that now. Now let's talk about active transport. Like we said earlier on, that this active transport requires energy, and the energy is in the form of ATP. The energy is in the form of ATP. That's the energy that the cells recognize. Your body cell can only use ATP, adenosine triphosphate. So, but it also requires specialized carrier proteins too. So these proteins are called pumps. Take for instance, we have sodium potassium pump is a form of active transport and it uses ATP, and this maintains the balance of sodium and potassium ion both in the intracellular fluid compartment and the extracellular fluid compartment. Okay, now we move to the organelles that are inside the cell. Let's start from the nucleus. The nucleus has a nuclear membrane, or you can say it has a nuclear envelope, and this nuclear membrane too is a double membrane. Then also in the nucleus we have DNA, deoxyribonucleic acid, DNA. So in the nucleus we have the nuclear envelope, which is a double membrane. We have the DNA, and the DNA controls metabolism, protein synthesis. Then we have the nucleolus. The nucleolus, what it does is to assembles ribosomes. So the implication of that is that we have in the cell DNA, we have we have in the nucleus DNA, then we also have RNA in the nucleus. But the nucleolus is the one that assembles the RNA. If you still remember, I think I taught your class the process of protein synthesis, transcription, and translation. But don't worry, we will still talk about that. Now, so let's go to the next organelle, mitochondria. See, this mitochondria is responsible for cellular respiration. So what it does, we call it the powerhouse of the cell, and the reason why it's called the powerhouse of the cell is that it produces energy in form of ATP. It completes the cellular respiration, so it produces energy in form of ATP. And these mitochondria, they are always abundant they are always abundant in cells with high energy demands, like the liver, your muscle. Your muscle also do a lot of work. So mitochondria, the muscles are rich in mitochondria so that they can have energy. Your movement is partly because of your muscle and your skeletal system. Then the sperm too, because the sperm has to travel in the birth canal, it has to travel in the female reproductive tract, and the distance is not small. So the sperm too is also very rich in mitochondria. So ribosomes, what do the ribosomes do? Ribosomes are composed of RNA, ribonucleic acid, and protein, and ribosomes are for protein synthesis. You can find them free in the cytoplasm, or you can find them on the outer surface of the nuclear membrane, or you can also find them on rough endoplasmic reticulum. What they are still doing is the function of protein synthesis. Endoplasmic reticulum, let's talk about it. There are two types of endoplasmic reticulum. We have the rough endoplasmic reticulum. The rough is rough because there are ribosomes around it, and these ribosomes produce protein for export out of the cell. Any protein that will be exported out of the cell will be produced by ribosomes on the rough endoplasmic reticulum, while any protein that will be used inside the cell will be produced by free ribosomes that are free. They are not attached to anything. Okay, so the smooth endoplasmic reticulum is for lipid synthesis, the synthesis of steroids, and for detoxification. Most of your drugs are detoxified in the cell, in the liver, by smooth endoplasmic reticulum. So do not forget this. It's an important information. We will come across it in one of the MPC anatomy past questions. The smooth endoplasmic reticulum is responsible for detoxification because it contains some enzymes that detoxifies drug. So what does Golgi apparatus do? What Golgi apparatus do is to, when your ribosomes on the rough endoplasmic reticulum produce protein which are to be exported out of the cell, The Golgi apparatus will now package them for shipment. So it's the role of the Golgi apparatus is this. Take for instance, you ordered something on Jumia. Okay, Jumia will prompt the vendor that so person has ordered so thing. They send it to Jumia. Jumia packages the product. So the process of packaging is what the Golgi apparatus does. Lysosomes. So what are lysosomes and what do they do? Lysosomes they digest worn out organelles and foreign materials. They contain enzymes that digest organelles that are worn out and foreign materials. Your lysosome in the white blood cells digest foreign material. So it's also important. Let's talk about the cytoskeleton. Just like in the human skeleton, the cytoskeleton is also responsible for the shape the cell form. The microfilament contains actin fibers and it's responsible for the cell shape. Microtubules are responsible for the movement of organelles and chromosomes. The movement of chromosome is important during cell division. Then we have intermediate filament. Intermediate filament are for structural support. Okay, so now we also have cell extensions. And these cell extensions, they are just projections from the plasma membrane. And they are mainly composed of microtubules. They are used for movement most of the time. We have microvilli. Microvilli are just tiny projections containing microfilament, and what they do is to increase the surface area of certain cells to enhance absorption of nutrients. Take for instance, in your small intestine has a lot of microvilli, and the function of this is for absorption. So when you eat, absorption majorly takes place in the small intestine because of the microvilli. It has a larger surface area because of the microvilli. Then we also have cilia. Cilia are microscopic hair-like projections, and they contain microtubules. In the respiratory tract, we have cilia because the cilia helps you to sweep foreign particles. The mucus will trap foreign particles. When you inhale, if the foreign particles escape the nasal cavity and gets into the upper respiratory tract or lower respiratory tract, mucus will trap them. So, and the cilia will now move this mucus into your upper respiratory tract, and you can either spit it out or take it in through your digestive tract. So we have flagella too. Flagella too also are made of microtubules, and they are primarily found on sperm cells. So we are almost coming to the end of the cell. The next thing to talk about is the cell cycle. When a cell runs out, it is replaced by a new cell. Although there are cells that don't replicate. But when a cell is old, it dies automatically. And this process of death is called apoptosis. Apoptosis means programmed cell death. When a cell ages, it self-destructs. It will die by itself because it has been programmed to die, and it will now be removed by the process of phagocytosis. So self-destruct is a common word. Some pastors used to use it. They will say, the way you are going, you will self-destruct. Self-destruct means that the way you are living your life, you will eventually kill yourself. That's what self-destruct means. It means that you will program your own death. So this is how cells in the body also die. Apoptosis, programmed cell death. Normally, that's how they die, except there is now injury or inflammation. Okay. So now in the cells with 46 chromosomes divide by mitosis, while the cells with 23 chromosomes, divide by meiosis. Now, apart from your gametes, your sex cells, the sperm in men and the ovum in female, all other cells of your body divide by the process of mitosis. But these sex cells divide by the process of meiosis. This is the reason why your somatic cells, there are two types of cells in the body. We have the somatic cell, which has 46 chromosomes. Then we have the sex cells. The sex cells are also called gametes. And these ones have 23 chromosomes. They have 23 chromosomes. So these somatic cells divide by mitosis, while these sex cells divide by meiosis. And this is what happened when they divide by mitosis. They produce two. Let's talk about the phases of the cell cycle. That's mitosis. There are two main phases. We have the mitosis, that's the M phase, and we have the interphase. These are the two major phases of mitosis. So in the cell cycle, we have these three that I have highlighted will be the interphase, while this one will be the mitotic phase. These three are the interphase. So what happens at the interphase? In G1 phase, the cell grows. The cell is just growing. That's what happens in the G1 phase. In the S phase, the DNA replicates. That's what happens in the S phase. The DNA replicates. You can also say the new DNA is synthesized. Okay, so that's why it's probably called the S phase. Then after the DNA has replicated, this is what happens when the DNA replicates. Let me explain so that you get the full gist. We have 46 chromosomes. Not here. Let's have it here. We have I need to use the ruler then. Okay, so let's see. In a normal cell, we have 46 chromosomes. So when the DNA replicates, the chromosomes will now be 92 chromosomes. This is what happens, 92 chromosomes. So after this happens, the cell will also enter the G2 phase. G2 phase means that it will now be preparing for cell division. That is what happens in the G2 phase. Then after the G2 phase, it enters the mitotic phase, the M phase. And the M phase is made up of majorly four. The last phase is when the cell now divides into two, which we refer to as cytokinesis. But the major four phases are what I like to use this mnemonic for. PMAT. PMAT. That's the mnemonic we are going to use for these four phases. The PMAT. The P, the first phase for M phase is the prophase, followed by metaphase, followed by anaphase, and followed by telophase. So we have PMAT. So let's take a look of what. Then the last phase is now cytokinesis. Cyto, anytime you see the word cyto, it means cell. That's what it means. So cytokinesis means that the cell will now split into two. So you see, in a cell that is not dividing, a cell that is not ready for division, the chromosomes are called chromatid. But when the cell is ready for division, you now call them chromosomes. So what happens in the prophase? In the prophase, the chromosomes will condense and the nuclear envelope will break, or it will open. The nuclear envelope opens. The chromosomes are inside the nucleus. So, but they must come out during cell division. So the major thing that happens in prophase is that the nuclear envelope breaks down, or it opens up. so that we align at the center rather, sorry. In metaphase, the major thing that happens is that the chromosomes align at the center. We call the center the spindle. So the metaphase, the chromosomes align at the center. So what happens at the anaphase? Why are we using chromatids here? Okay, so the chromosomes we now, they have aligned at the center. The chromosome will now separate, move to the opposite ends of the cell. Okay, so when the chromosome moves to the opposite end of the cell at telophase, the nuclear envelope now reappears. The nuclear envelope that had broken down in prophase will now reappear at telophase. Once the nuclear envelope reappears, that marks the end of the mitosis. Then this, okay, so you see this is prophase, and these are the chromosomes. Then this is the nuclear envelope. Anytime you see purple, that's the nuclear envelope. In prophase, this nuclear envelope will break down. It has to break down. Okay, when it breaks down, then the chromosomes will now align at the center. That's what happens in metaphase. The chromosome align at the center. In prophase, this purple has to break down. You will see that this purple membrane did not follow this chromosome to this phase. You can see clearly, right, that the purple membrane did not follow the chromosome here. So what happens in prophase is that the nuclear membrane breaks down. Then when the nuclear membrane breaks down, in metaphase, the chromosomes will now align at the center. So after aligning at the center, in the anaphase, the mitotic spindle will now separate them. It will pull them. The reason why it has to pull them here is that because this cell has to divide at the center. So complete chromosome must be here. Complete chromosome must be here. So they have to pull to the opposite end. Okay. So when they pull to the opposite end, at telophase, the purple that is missing now reappears and envelope the chromosome on this side. It also envelopes the chromosome on this side. Then when this is done, the cell now divides at the center and form two daughter cells. Please, you can unmute. If you don't understand this thing, I will re-explain again. Please unmute. I need you to unmute and give me feedback if you understand what I have just explained. Do you understand what I have just explained?
Yes.
All right. So very good. Let me go all over again. At prophase, the chromosomes have already replicated. Okay. So at prophase, this purple, which is the nuclear envelope covering the chromosome, has to disappear, or it has to break down for the chromosome to be released. So this is what happens. Yeah, so it breaks down. Here, the chromosomes will now align at the center. Don't worry, I'm going to send these notes to you. But the chromosome will now align at the center. These ones that you are seeing here, they are mitotic spindle. We call them mitotic spindle. They hold the chromosome at the centromere. But I don't want to complicate it so that you will get the major point of mitosis, because year in, year out, students don't used to understand this mitosis. So I have tried to remove all the extra explanation and just go straight to the point. So at the metaphase, the chromosomes align at the center. The replicated chromosome, you will see that this chromosome is 2, blue 2. This is pink 2. This is green 2. They align at the center. Then in anaphase, they have to move to the opposite end. So here they separate and move to the opposite end of the cell. When this is done, let me start this explanation again. You see this yellow stuff, they are mitotic spindle. So they appear in prophase. Then that's the first thing that happens. They appear in prophase. Then the nuclear envelope breaks down. So when they appear in metaphase, they will now join the chromosomes at the center. The center is called the centromere. So, and the chromosome will align at the center. So in anaphase, the chromosomes will be pulled to the opposite ends. Then in telophase, nuclear envelope will appear. Then you will see that at telophase, this mitotic spindle, they have almost disappeared at telophase. The yellow like cobweb, they have almost disappeared. That's what we call the mitotic spindle. So then the nuclear envelope reappears. Then the cell now splits into two. Cyto means cell, kinesis means splitting. So cytokinesis now happens, the cell splits into two. So that is what marks the end of mitosis. Now, so we have come to the end of cell and cell division. So you can unmute your mic. We will take some questions. You see, the question we want to take now, they are very simple questions. But I have close to 100 questions on cell that are complex, meaning that these are the kind of, not too complex anyway, but these are the kind of questions you should expect in your MPC anatomy. So, but for the sake of clarity, we will take just these few simple ones. So unmute, please. Let's take questions. But while you're on mute, if there are aspects of the presentation that you don't understand, tell me now so that I can go over again. But if you understand everything, no problem. But if there are aspects of the presentation that you don't understand, tell me now so I can go over. Okay? I know you will not say anything. That's not a problem. Even if you don't, please, if you are on the road, you can mute your mic. We don't need you. If you are on the road, mute your mic. Let the people that are in their room talk to us. What is the smallest functional unit of the body? Cells. Almost all of you will be able to answer this question. The human body originates from a single cell called the zygote. The plasma membrane is primarily composed of phospholipid bilayer. That's the major thing. Phospholipid bilayer. Which of the following is not a function of membrane proteins? Which of the following is not a function of membrane proteins? Which of the following is not a function of membrane proteins?
DNA replication.
DNA replication, very good. What is the main function of the plasma membrane? What is the main function of the plasma membrane? What is the main function of the plasma membrane? Selective permeability. Yeah, they allow some substances to move while they stop some from moving. What type of transport does not require energy? What type of transport does? Passive transport. Diffusion involves the movement of. Thank you very much. The diffusion involves the movement of molecules from high to low concentration. Osmosis is specifically the movement of water molecules. Water. Active transport requires which of the following?
Energy from the